Thank you so much, Manuel, and thank you for a wonderful talk, David. Just for the record, I have 8 cores on this laptop, so you should cut that time in half. I'm going to be talking about where we are with informatics infrastructure, and I'm going to try and project where we're going. I was here at the beginning in 2005 at the Natural History Museum at the first meeting of the Barcode of Life community, and there were about 50 of us there, and it's just been fantastic growth. And I'm going to try and take a look back and look at the factors for success, the infrastructure that helped us get there, and how we decide to move forward to scale up and continue to grow. In 10 years, we've achieved a lot with 4 million barcode records. As Paul mentioned on Tuesday, over 500,000 species barcoded, and so many different nations represented. And from an informatics standpoint, I think there are 4 key things that have allowed us to do this. Central to that has been data standards. The data standards have joined us in how we collect data, what we collect, and setting minimum quality for that. Secondarily, community databases, and of course the people, the collaborative networks, and the software tools. I don't think these can be separated from the informatics infrastructure. The data standards, again, have been critical, and the Consortium for the Barcode of Life has been critical to helping make that happen with community consultation and capacity building. They focused on quality and completeness of data records, and NCBI was another important player dealing with enforcement of provenance and locus standards. But in the biological community and the scientific community, standards get set up, but we find that you need infrastructure to help implement them. Otherwise, there are challenges for researchers to comply with these standards, and BOLD has been central in that. To date, the majority of barcode records available are either born in BOLD or have been mined into BOLD. And we can see the growth of that over time from 2004. 2005 was when it was first made publicly available, and it's continued this exponential growth of the data in number of barcode records. But the number of species records have continued to grow as well. We hit a bump at about 2012, where we came to the realization that a secondary taxonomic impediment was there, where our access to taxonomic information, the rate at which we could add taxonomic knowledge to this database was limited. And that's where bins started to come in, a way of demarking the space of where the biodiversity lives based on sequence data. Now, our early community decision to use a single database, or the majority of us anyway, to put our data in a system like BOLD, even before it's public, has been incredibly useful. One is we can do things like this, where we can measure progress before data is published. These are community benchmarks based on completeness in key groups. Each circle represents the number of species that are available, and some have greater completion. On the geographic axis, we've got Lepidoptera of North America, birds of Argentina that have done incredibly well. Fish on the taxonomic side have done well, so have birds. But other groups need support, need effort, need communities to take action. But the other aspect of this is having all this data in one place has allowed us to reduce the duplication of effort. When we work in silos, we don't know what others are working on, and we can ensure that our resources are properly expended. After the database come the collaborative networks, and I look at the growth in users in BOLD over time. In 2005, we just had 102 people. And these are just a few representative I picked from people who are attending the conference that were there at the beginning, and now well over 14,000 users. And one of the things BOLD does is allows these users to share data with each other. to break down these silos so that even in a private space, you can work together. And this is where we are now in 2015, where each line connects an institution across 94 countries. Each line on this figure represents a sharing of 1,000 records, with the yellow representing 1 to 10,000 records, and blue 10 to 100,000 records, and red beyond. And it's just fantastic because if you look beneath the line over there, this is the amount of sharing that happens within a nation. And it's nowhere near what happens across nations. This is a truly global effort where researchers across institutions and nations are connecting and sharing data and refining to ensure that we have the best and highest fitness for use for the industry and for other researchers. Now, the main nations are the ones that were involved early on. Costa Rica is an outlier in there, which has been wonderful to have that diversity in there, sharing over 100,000 records. But when you look at the second level, 10,000 to 100,000, many nations rise to the surface. And across continents, across GDP scales, and 66 countries from every continent is also represented here, sharing data. And what we see is nations rise with the amount of data they contribute and share, because biodiversity does not respect national boundaries. So we've done a wonderful job in increasing the size, the quantity, the volume of the database. But we need to look at the efficacy of the database, and I think we need to call that more depth than breadth, because capturing species just very broadly and blindly makes it very difficult to ensure that you're capturing all the diversity out there, because it's a multidimensional space. So we start by looking at animals because it's a bulk of the database. Animal barcoding was the first community to start building the database, and a large amount of the diversity out there is represented by animals. So we have about 400,000 species, 163 of them with full taxonomy identified down to a Linnaean name, but about 500 orders. So a good span across the taxonomic space. So to look at the depth of this, we can do a simple statistical test, which I did, taking 4,000 species from 200 orders, but limiting it to 20 species per order, because there is definitely bias in the database based on strategic plans and economic relevance of different taxa. And what we can do is we can present these species on a plane, Cartesian plane will do, and we expect for barcoding to work that butterflies and owls are going to be in different parts of that plane, and we can put these queries on that space and say what's near it, and look at these concentric circles of proximity. And then we can plot that through time. Because BOLD has been around for a long time and we make very consistent backups, I was able to go back in time and reproduce the identification engines from 2005, 2006, all the way up to 2014, and look at how these 4,000 species would have fared over time. And there are 4 thresholds. I know we've moved past thresholds, but they serve a good purpose for a simple statistical test. 4 thresholds that serve as proxies. 98% is a good proxy for you're more likely to get species. 95% you're in the genus region. 92% similarity. You could get to family, and 90% around the order level. And what we find is that we did really badly in 2005, but we've moved the mark considerably. Now, you'd expect in 2015, with the test data coming from the library, to get much closer to 100%. But for this to be a good test, you have to remove the test data from the reference library. And what this shows is that 25%, or more than 25%, of the test data is singletons. So at 98%, we could not find anything. So the long tail is a big challenge for us in building this library. But there's hope, because the other marks, the other lines, are considerably higher, especially the top line around 90%. So why is this important? Because if we can place things to order and family, as Paul was saying earlier this week, we can access the traits of those groups. If you know that something's in the family of Culicidae, you know it's a mosquito, it could be a vector for disease, and you can do something about it, even if you don't have a species name. So distances are not the only way that we can start to put these unknowns into these higher level categories. We have other features because of the data that we've collected. So two features, just on a quick blush, is amino acids. Fortunately, with animals, we've got a protein coding gene that gives us another layer of information. So we could take the amino acids and run a principal component analysis and across major orders of insects, and we find that there is structure in the data. We find that in a lot of these cases, we can put it into an order based on these groupings. So, but it's not perfect. And then we can go over to using k-mers. K-mers are short words extracted from the sequence, and we find that there are other groupings and other strengths. Some things are difficult. If you look at Hymenoptera, these rate-accelerated haploid-diploid groups, they occupy a ton of space on this hyperplane. But there are other features that we can look at. There's work to be done here. The database is still reasonably untapped in the features we can get. So moving on from the library of the known to the unknown, the barcode index number has been an important tool in helping us catalog the diversity that's out there. And it's really two pieces. One is that it's an algorithm, a clustering algorithm, that takes sequence data and puts it into OTUs. And that's an interesting point of discussion. There are a range of algorithms, but I'd say that's not the most important part, because algorithms evolve and change and get better. The most important part of the system is the registry, that when we get an OTU in a place and time, we register it as occurrence. So it's a new kind of occurrence, that this cluster appeared at this location instead of just the species appeared at this location. And then we can aggregate all the information we have about it, the morphological characteristics, the association with other organisms around that entity, so that it's useful. And because it's registered, it's reusable across studies. There are many studies out there that generate OTUs, and those OTUs end up living in the publications with the associated metadata. We want to capture that and database it so that we can do these longitudinal analyses and cross-cutting meta-analyses. So I'll give you one example. These bins aren't very human-friendly, but AAF2716 popped up around 2011 in these 3 different sites with 3 different names. It was in Costa Rica under Horatia Jansen 03, which is very typical because we make sure that when it's an interim name that it looks nothing like a species name. And the BioBus, which you've seen here at the Biodiversity Institute of Ontario on campus, captured one named Nodioplusia illustrata, which actually turns out to be the true name. And in Australia, Tinoplusia sp. Anic 1. And the story with this particular moth is that it was introduced to Australia to control this lantana, beautiful plant though, but an invasive and a pest. But it sat in the CSIRO collection without a name, and bins were able to connect this together. These 3 names don't exist anymore in bold because it has been reconciled with a system like bin connecting all these together. And there are many more stories like this. We can do continental scale comparisons. So in fact, if you take a 7-continent grouping, we have about 19,000 bins that occupy more than one continent. And I can't emphasize the importance of registering these bins. And amusingly enough, even in mammals, there's a little bit there where bins capture some of the diversity. And another example of how these bins are useful, especially in the context of climate change, is the latitudinal range of the large number of these bins that occupy such a latitudinal range is quite surprising. So the test of endemism can be done with bins, and with the scale out of malaise traps and non-human collections, sort of robots running around with nets, if you will, this is going to become incredibly powerful for biomonitoring and risk assessments. So I'll move off of that. At the beginning, when a bunch of us got together with the barcoding concept, I suspect most scientists are science fiction fans, and who doesn't love Spock? And the tricorder that he seems to be the only one capable of using. And there was an article in Boing recently that the technology that links taxonomy and Star Trek is this tricorder. But they pointed out something really important. It wasn't the device. It wasn't the shape of the device. It wasn't the technology of the device. It was what it did, which was give someone who could not use their eyes, ears, taste, touch, to get access to information. That's really what we're about. And the Eyeball project started with this hope, this vision, that we wanted to allow anyone, anywhere, to identify any organism. Because when you can identify the organism, you know how it affects you, how important it is, and that you need to make a decision. So we've been waiting for the device, but in 2013, we realized that we could take action. The library was powerful enough. We could take action without a device and preempt the device and build a community of users that demanded the device. And so the LifeScanner project started. And it started in partnership with a large software company, SAP, that's been helping us and supporting us with this initiative, which is use the technology at hand. We use mobile phones. Smartphones are quite powerful these days. And we hijacked the personal genomics technologies. 23andMe is one of the most popular personal genomic kits. You can order it online, and you spit in a tube, and you throw it in the mail. We use the exact same mechanism. We've developed reagents that allow for safe transport of these samples. And you record it, and you throw it in the mail. It goes to a lab, and back comes the information to your phone. You don't need to be on board. You don't need to be a biologist. And also, you get the descriptive information, the distributional information. Access to information is at the root of this, so that you can make a decision, so that you can appreciate biodiversity. The process is very straightforward. You capture the additional metadata around the specimen that's tied to that individual record, throw it in the mail, and we're building partner labs to process these samples. The Canadian Centre for DNA Barcoding here has been an incredible partner in accepting all of these, but we're building labs, partnerships with labs in Europe and the US to make this happen. But it's all centralized so that the user does not have to talk to the lab. The sample finds its way to the lab. So I just want to take the opportunity to show you some of the material that we have. But first, we've generated a video to try and explain this project, and if you'll indulge me, I'll just play it for you.
Life on our planet is incredibly diverse and complex. But where does this diversity and complexity come from? How does a caterpillar know to eat leaves? The answer is DNA, Life's Instruction Manual, written in a language based on 4 molecules called nucleotides, referred to as A, C, T, and G. These occur in pairs in DNA. There are billions of these pairs, and it is the specific arrangement of these pairs that form the instructions for an organism, its genome. LifeScanner uses a method called DNA barcoding to analyze a small piece of tissue from a specimen, isolating a short segment of its genome to identify it. How does LifeScanner work? Simply collect a specimen with our collection kit, use the LifeScanner app to record important information about it, and send the completed kit to us. When your specimen arrives at one of our partner labs, it is given a unique ID number and linked to you. You can then use the LifeScanner app to track its journey through the DNA barcoding identification process. First, a small portion of tissue is taken, and your specimen's DNA is isolated and extracted from the cells. Only a small part of this DNA is necessary for species identification, the DNA barcode. But we need to make many copies of this segment to be able to read it. We use a technique called PCR to exponentially increase the number of copies of our target segment of DNA, the DNA barcode. This gives us sufficient genetic material to use in the next step, DNA sequencing. To prepare the DNA for sequencing, we create a mixture of the PCR-copied DNA, regular A's, C's, T's, and G's, and special fluorescently tagged A's, C's, T's, and G's. DNA polymerase replicates the input DNA with regular nucleotides and incorporates fluorescent nucleotides at random positions, creating DNA fragments of different sizes and ending at every possible position in the target segment. A special machine then analyzes the prepared sample by reading the fluorescent nucleotides in order of size. The machine produces a long string of A, T, C, and G characters. The arrangement is converted to a digital sequence that is unique for each species and is between 500 to 600 characters long. This sequence is the DNA barcode for your specimen and can be used to identify it. Computer software is used to compare your specimen's barcode to a global species database to determine the closest match to your specimen's sequence. Once a match is made, you will then receive details about the species you collected. The data you collect with LifeScanner and the DNA barcode obtained from your specimen will be submitted to the International Barcode of Life project and added to a global scientific database. These DNA barcodes will help researchers around the world study these species further. Become a citizen scientist and contribute to the scientific record. Order the LifeScanner DNA Species Identification Kit to identify organisms around you.
So I know what some of you are thinking, that we're going to get this. And I'm sure there are going to be some of these, but I think the world is changing. Just 2 days ago, very great, good timing, an editorial in Nature, The Rise of the Citizen Scientist, and they identified two things. One, science is broadly accessible now, and the modularization and mobilization of technological tools is really what's making this possible. So I think we'll have a positive result. And the kits that we've been getting back have certainly been a testament to that. Obviously, you get some of the unusual things of, someone, I don't know who's eating boneless turtle. The bone's the good part, or alligator meat, but we also get some interesting things. We get new species to the BOLD database. So these are groups that have been over on the, whoops, that's where this ends. Over on the left, my left, your right, are new marine species that a National Geographic group have picked up with the LifeScanner kit, and they've become a regular user now, and this streams in. The little insect over there was eating, in fact, that's the only contribution that I've made so far, was eating one of the plants in my backyard, and I couldn't get an identification, and I did, and I was able to take action that was environmentally friendly instead of broadly spraying. So supporting these decisions is really important. And Manuel, I hope I'm not going over time. Oh, good, because I'm only halfway through. So I'll try and rush through the rest of this. Looking forward, this is where we've come in 10 years, and I'm really excited about the next 10 years. So we're moving into the second decade. How do we need to change our mindset about data? And it's really to embrace big data, and there are two aspects to big data. One is the dimensionality and the volume. We need to collect a lot of data, but we need to add a lot more columns, because the power of big data is becoming apparent. in the impact that it can make. So far, we've been able to get to the, whoops, what happened twice. What's happening, what's happened in the past, but we can't really tell what's happening the way we can with weather. We could have predicted that it was going to rain out there, and in fact, my colleague Derek Tittensor is going to be talking about that next. But we need to build the data infrastructure to allow for this to happen. The other thing is we need to consider the multiple scales that we work at, and there's probably another scale past that. We started off at the range of 10 to the 2 with our collecting efforts and moved up. But on the far left with strategies like Malay straps, it's an example, it's a concept. There are many other strategies that are automated, and that's what's going to drive the volume of data that we need. And that matched with NGS-based barcoding is going to crack this discipline wide open. And we need to support these. So we're starting to do that with Bold 4. That'll be launching on September 29th. And it takes steps towards addressing some of these challenges. So the first thing, we need community-curated databases. The problem with a system like Bold, with everyone pushing in data, is you get noise and signal. We've done this already. We worked on the project with U.S. Food and Drug Administration's Center for Veterinary Medicine, and they were able to construct a library within Bold and have an identification engine for that. So Bold 4 will have this, where you can convert a data set into a closed identification engine and build curated libraries and publish them so that other people can use them instead of using the whole database. So this is going to make this kind of analysis a lot easier. It also is going to help expose some of the gaps in the library. So this is a 3-tier library with tier 1 being highly curated. They've got a definition of dirty 22, things you definitely don't want to find in food, like ringworm. And they had great coverage, 78% of that. But as you go down the list, the coverage changes. But these are risk factors associated with using this as a diagnostic tool. Next is we're adding community-defined metadata extension capabilities. The barcode data elements have been fixed for 10 years from the original data standard. We need to expand these standards to include the types of data necessary for forecasting. So this is just one example where we're looking at a study from 2011 where we were looking at host-parasite relationships. But the Biodiversity Institute of Ontario's collection staff has generated body mass for over 20,000 different Lepidoptera and looked at that distribution. And that's now a column in the database. And you can continue to add additional columns. So Bold starts being broadly useful. We'll keep building new analytical tools. Bold 4 will have a few more. But there's a challenge here, which is you can never make enough analytical tools that meet the broad community's requirement. So we're doing something very different with Bold 4. We're going to let you build your own tools in Bold. So we have a plugin framework based on the R language, and R is a fantastic solution in the scientific community with over 6,000 analytical packages. I can't compete with that. My team can't compete with that. So why not embrace it and let the community define what these are? So the process will be you upload your R script, we evaluate it. We'll be sort of like the iTunes store. We can't let you post just anything. It's got to be secure and useful. And then other people can utilize it. And I've had some dialogue with the GigaScience Journal where they feel that if a tool is used often enough, they will provide the means for a 2-page application note because of heavy use of the tool, and it can be cited. Additionally, we'll do something else different because mashups provide a way to solve different problems. We'll support analytical pipelines. This is a very simple analytical pipeline that I know everyone uses. You basically drag these nodes onto the page, and then you connect them. So this is a common starting point for any researcher doing molecular work. You start with the records that you have, and you go and search for things that are within its vicinity, within a certain molecular distance, and then you bring it all together. And so this does it with a push of a button. You define the parameters, and off it goes, and you have a data set that combines the data you've generated plus all the public data and all the shared data that you have access to into a single data set for analysis and study. And it's early days for this. We'll only support simple processes initially, but I'm really excited about how the community can drive the pipelines that are going to solve this. And finally, again, because the community has expanded so widely, we have to find partners, and an important partner for us has been SAP. There's been demand for a desktop tool from BOLD for quite some time, and I think SAP's Lumira will be the solution. So SAP Lumira is a big data analytics desktop tool, lets you explore massive amounts of data, and the discussions we've had with SAP will allow us to develop visualizations inside the Lumira platform, and we can connect to BOLD and pull data down, and then you can analyze and visualize and explore that data on a desktop platform. So incredibly powerful way of working with large amounts of data. It'll handle millions of rows. So I'm really excited about what people are going to do with that and how we can add that as a layer of infrastructure moving forward. And SAP's Lumira will be free to the community and can be used, the figures can be used in publications. Manuel, how am I doing on time? I'm done. Okay. I should have looked in that direction. Finally, the boring stuff. Some additional features. Checklists. We're really supporting synonymy and progress tracking with checklists and shopping lists for taxa. A data portal for core facilities so that they can manage their data better and funnel it to researchers. Complete record histories, and also something that's been interesting is we're making the RESL algorithm open to researchers on BOLD, so you can take a data set and you can run it just on that data so that you can use it in your comparative analyses. We're going to add ABGD, GMYC, and the full range of OTU generators so that you can be as pluralistic with your approach as possible. Additionally, and probably of major interest, is we're going to support the storage of NGS data post-processing. So this is where you've clustered it down to 10,000 or less sequences. They won't end up in the ID engine, but then you can utilize a lot of the tools that BOLD has. And NGS, use of NGS has spread from dealing with old material to biomonitoring, and so we want to try and support that. Last one, we recently received funding to build a new platform that we're calling mBrave, Meta-Barcoding Research and Visualization Environment, to support, to build something like BOLD for those researchers that are using NGS for barcoding. We're not trying to reinvent the wheel. We're going to utilize our partners. SAP has great expertise in big data systems. Build on what we can get from Galaxy, utilize ENA and NCBI's short read archive, but most importantly, adopt the standards of the Genomic Standards Consortium. There have been wonderful standards that are cross-cutting, and extend those standards in consultation with the broader community so that we can start capturing the metadata that we need to around these NGS data sets. I won't wrap up. I'll take my last minute to thank people because I really wanted to do this. I have to thank my fantastic team, the BOLD team. I hope they'll stand up if they're here. I just want to give them a hand. None of this is possible without them. So guys, just stand up for a second. And of course, my mentors, Paul Hebert and Dan and Scott and many others here that are listed that I can't list, and my colleagues at the Biodiversity Institute of Ontario. Manuel, I'm so sorry.
Thank you very much, Ivan.